Dr. Ajit Samal is a professor in computational biology group at Indian Institute of Mathematical Science, Chennai. His primary research interest area are in network sciences and computational systems biology. He is also the head of Max Planck Partner Group in collaboration with Max Planck Institute for Mathematical Sciences, Leipzig, Germany. His research group has developed a phytochemical atlas, the largest digital resource to harness knowledge on traditional Indian medicinal plants for aiding ongoing research efforts in drug discovery and human well-being. Let's hear from more details on that.
Okay.
For the lecture, sir, we would like to present a small token of appreciation. And may I request kindly Professor Mita to present a small memento to Ajit sir. Thank you, Ajit.
Thanks, Pita. As always. Yeah, thank you. Okay, so let me start by thanking Smita and everyone for the opportunity to present our work here. It's been recorded, so let me just put it on record that we created this resource, but many of the stakeholders recognized that my group created this resource after Smita gave me an opportunity to present at a conference. So while people use it, many people don't relate it to the Institute of Mathematical Sciences, and it really changed with Smita giving me opportunity and one of the conferences she organized here in IIT Madras. So yeah, so just a disclaimer that I many people think that I'm a jack of all trades and master of none, and maybe it's true. Uh, Primarily because I started my lab about a decade ago, and all the research that I have been doing in my lab for 80% of the time, they had no correlation or relationship with what I had done before in my PhD and postdoctoral research. So, just to give you an overview of the research, how it is divided in my group currently. So, the major area in the last 7 to 8 years is what we call as chemical spaces for drug discovery, systems toxicology, and human exposome. And the work that I'll be presenting today essentially falls under one of the threads of two sub threads in this area. I have core expertise on development of mathematical computational tools for analyzing networks. So during my PhD, I was trained to do constraint-based modeling and also Boolean modeling, the kind of things, Maya was talking in the previous lecture. And in my group, subsequently, what we do is we have been developing tools, particularly for analyzing networks and higher order networks. And further, we continue to do a bit of Boolean modeling as well. One of the interesting areas that I am particularly, passionate about is basically systems modeling of metabolism and protein secretion system in microbes. There has been some amount of work here also, but hopefully in the next couple of years, you will hear much more of our work here. So these are three areas where we lead projects. That is my group, sort of takes a lead in these projects. Uh, we are in a purely theoretical institute, but I do collaborate with experimentalists, for example, with Smita and many others. And in some of these projects of the experimental colleagues, we do play an enabling role as well, where we analyze their omics data, in particular in the context of networks, and provide new hypotheses or understanding to the organism of interest or biological processes they may be studying. In particular, in the last few years, uh, these kinds of enabling role projects have particularly focused on basically people who are working both on the pathogenic side of Mycobacterium tuberculosis and also on the host side. So with that brief introduction to the research which goes on in my lab, I'll go on to the topic of today's research. Although it may seem disconnected from many of the other things I do, uh, essentially the topic I'll talk about is broadly what I consider in my lab area, which I call as compilation, curation, and computational exploration of the chemical spaces. And in particular, we have been focused on two types of chemical spaces. One, natural products, which are potentially beneficial to the humankind. And to this audience, I don't need to make a case for that. And second, environmental chemical spaces, which could be potentially harmful to humankind. And in this first area, I will particularly focus on our work to essentially compile the largest resource on the phytochemical content of Indian medicinal plants, which is called impact. I must mention that while this is what we may be recognized more in the Indian space, comparatively much more work we have done in this space in the last few years. So, when I started doing this work, it was very serendipitous, and our primary motivation was to create a natural product space for potential drug discovery. So, I don't need to make a case to the audience here. Natural products, of course, continue to be immense interest towards scientists working in the field of drug discovery. So, we have an unpublished data set in my group where we have actually mapped the history of all the drugs, small molecule drugs, which are approved by FDA starting from its inception in 1905. It's unpublished data, and what we find that as of today, that is 2023, most of the small molecule drugs, almost 40% of them are either natural products or started out from a natural product based lead molecule. So, while there was a certain dip at some point in the history of drug discovery where the drug discovery focused more on the synthetic molecules, we see in that curve. I'm sorry I didn't put that slide up here. That again, there is a resurgence in the interest in looking at natural products for potential drug discovery. So essentially, we are interested in the subspace of the chemical space, which is biologically relevant, and within that, the natural product space. So, our primary motivation to do this work will be summarized in this slide. As all of you are aware of, plant secondary metabolites or phytochemicals, help the organism in various, processes. For example, plant defense that Maya was mentioning. Essentially, they help the organism to survive in their ecological niche. Also, plant extracts which contain these phytochemicals have been used to treat human ailments for centuries. I mean, this is not particular to Indian or Chinese civilization, but any civilization you go back in the history. And of course, many present day drugs, starting from morphine to paclitaxel, many others, they were initially isolated from plants. And in particular, when we started doing this work, it was important to us to recognize that various systems of traditional medicine, in particular Indian and Chinese medicine, they use these medicinal plants in different formulations to treat their diseases. So, essentially, what we noted is that when we started doing the work in 2016 is that if you look at the information on traditional Indian medicine, in particular the phytochemical content of various plants which are used, this information was largely buried in books. And this information was not also electronic in nature. And I argue that this non digital nature, essentially at that point, limits its effective use towards modern drug discovery. So, furthermore, what was stark was that when we did this work, in 2016, when we started, we noted that if you look at digital databases which enabled computational approach towards drug discovery based on natural products, if you look at, for example, Chinese herbs, there were several public databases which were much more larger compared to database on the phytochemical content of Indian herbs. And so essentially what we felt is that we need a database, and we started out with a database, but I think we need a computational platform which essentially enables traditional Indian knowledge and natural product based drug discovery. And with that in mind, we embarked on creating a resource called Impact. Uh, and we built the first version of Impact, which was published in 2018. Again, I want to emphasize to people here because this is a diverse audience. We set out our building this database from the perspective of drug discovery, but over the years, frequent to the first publication, we have had various feedback from several of the stakeholders, and that has helped us actually enhance, expand, and improve the database for various needs. And moving forward, actually, what we would like to do, and I won't be able to present today much, but we think that we can improve the database, for example, to uh, facilitate efforts in the country towards integrative medicine. So, what I will quickly walk you through is how we created this dataset and why this dataset is very valuable at this moment. So I must mention that in 2018 we published the first paper, and it was quite a bit of manual effort to create that resource. But subsequent to 2018, based on the feedback, we essentially worked, tirelessly for about 4 years and released the second version, which was released actually in 2022, exactly a few days before actually Smita organized a IoTech meeting here, and the paper was published in early 2023. So as Impact as it stands currently captures information on more than 4,000 Indian medicinal plants. We have a highly curated data set of about 18,000 phytochemicals, and we have captured the therapeutic uses of 4,010 plants, and the number of therapeutic uses we have is about 1,100. And it is clearly facilitating computational approaches towards traditional knowledge-based drug discovery. So what I plan to do over the next 5 minutes is quickly walk you through the process with which we created the first version, and then I will explain what we did to expand and enhance the first version to the second version. So the first starting point when we embarked on this journey was to create a list of Indian medicinal plants. So in the first version, we had a list of about roughly 5,000 Indian medicinal plants, which we compiled from published information by various sources, in particular by the Ministry of Ayush and the CSIR CMAP Institute. And once we had this list, I mean, this was some amount of work because these plant names were in synonymous forms. So we mapped them to their accepted names and we got a non-redundant list. And thereafter, we embarked on capturing the known phytochemical content of these plants. So the phytochemical content was captured from broadly three resources, and the major resource was books. And this is very important because most of the high quality information, at least to my knowledge, is in books and not in research papers currently. And so we captured this information from books, and some amount of natural language processing of the existing published literature was done. And we captured information from about 7,000 papers. And I must mention that we were not the first to think about this work. So before our work, there were two such efforts, at least to my knowledge. One is this database, which is called Phytochemical, which was created by my colleague Ganesh Paglar, who was then, when he created this database, he was at IHBT Palampur, and now he is at IIIT Delhi. He has created a resource called Phytochemical, which was for not five Indian medicinal plants, but actually these were Himalayan medicinal plants. And particular to Himalayan region, and they had 963 phytochemicals documented there. And this was not a database, but actually a data set which was published, but actually you don't find the data set as such as a supplementary material or any database. And this was a paper which was published, which had captured information for 295 Ayurvedic Indian medicinal plants and about 1830 or 1829 phytochemicals. So I won't bore you with how we grabbed this data out, but I mean, it was submitted to a database in Europe called NutriChem, and we extracted the data out of there. So, so what I want to like to mention is that uh, while, for example, Phytochemica had done out for only five Indian medicinal plants, and they had documented 963 phytochemicals, but if you look at the data set of the 963 phytochemicals, the list, the structure, they could associate the chemicals to the chemical structure of these phytochemicals for only roughly 500 of them. So roughly half of the phytochemicals in their database don't have an associated chemical structure. So actually, you don't know if it's an error or not. And I mean, we also attempted to map it to the structure, and it was not straightforward. So one of the fundamental principles we wanted to follow while we wanted to create impact was it should be computable. That is, if we list a phytochemical, it should have a clearly associated structure. And in impact 2, as I will tell, we actually resolve the stereochemistry to the best possible. So in our effort, what we did is we collected these names of phytochemicals which had been reported in various literature to be produced by various plants. And at this moment, I would like to emphasize one thing. We don't create any knowledge in terms of phytochemical content. We are only organizing knowledge. I hope it's clear. And Impact is also very particular about this because the onus of the correctness of the information in Impact doesn't lie with us, but actually the literature reference that we cite in the table. So if you are using Impact, please go back and look at the third column for which source from where we got the information, and you should attribute that information to there. Okay, so I mean, if you are particularly thinking of doing an experiment or something, you should go back to the source, see if you are confident enough to go forward or not. So so what we had was that we got for about 1,800 plants about 30,000 or 35,000 names of phytochemicals. And of course, these names were very synonymous. They were common names, and we had to associate it to the right structure. So what I wanted to say is that of the 30,000 plus common names that we had, we could only map 15,000 of the common names to uniquely to 9596 standard chemical identifiers. So you may ask, what about the remaining 30,000? Actually, they still remain like that. But in impact, you only see a phytochemical being listed if we could associate it to a clearly to a chemical structure. Okay. So there is still a large amount of things to already discover, because we have huge number of names of phytochemicals which are reported to be produced, but we couldn't still map to the structure in standard chemical databases, or those resources or the papers or the books have not given the chemical structure. Okay. So with that, I mean, briefly, it's a lot of manual work because we had to go through each data point manually. I mean, my students did it. I did nothing of those work. So basically, what we achieved was we got a data set initially for about 1,742 Indian medicinal plants and their 9596 phytochemicals. So, so since each of these phytochemicals had a clearly mapped 2D structure, you could do a lot of computational chemistry slash chem informatics with it. So we, of course, gathered the 2D structures. We either got the 3D structures from resources or could predict using tools. We could compute the physicochemical properties, predict the drug likeness, predict ADMET, predict human target proteins, and all that information was systematically compiled and put in the database in a very user-friendly manner for people to browse and use. So apart from the phytochemical content in the first version, what we did is that we also wanted to capture the ethnobotanical or therapeutic use information for these medicinal plants. And there we made a conscious choice not to rely on research articles, because that's a very contentious situation where you take any well-known Indian medicinal plant and put any well-known disease, somebody would have tried to relate them. Okay. So we didn't want to get such information. So we basically relied on very solid authoritative books, especially books by Ministry of Ayush, to gather the therapeutic information of these plants. Other than gathering the therapeutic information, one of the minor innovation that we did is that, of course, again, this therapeutic information is given in synonymous vocabulary. We mapped it to, terms which are standard in Western medicine, in particular to disease ontology, MESH and OMIM. Okay. So, and finally, now what about formulations? Okay, that's important for drug discovery. So there has been a large scale effort by CSIR with Ministry of Ayush called the TKDL. So they try to digitize the information on formulations, and TKDL um, has digitized 2.5 lakh formulations. Out of these 2.5 lakh formulations, only 1,200 are representatively present to be openly browsed on their website. We, of course, did not want to reinvent the wheel and get into that space. But what we did was that we tried to map if any of the plants which was in Impact 1 is listed in TKDL as well in a particular formulation, in particular to the 1,200. So the idea is to link the formulation to the plant, to the phytochemical content, and see if the plant part for formulation, if it's a single herb formulation or a multi-herb formulation. Subsequently, if this kind of data could help predict or explain potential therapeutic use which has been there for years in the culture. So that's essentially what we had in Impact 1. But it was just not a database, but we did a lot of cheminformatics analysis in the paper. I won't go into the details of the cheminformatics because similar analysis is what we did in Impact version 2 and go into details there. But just to repeat, the three different associations we had the medicinal plant and their phytochemicals. That was the centerpiece of our work. And then we had the medicinal plant and the therapeutic uses, and some information on medicinal plants and their use in traditional Indian medicinal formulations. And then we did three different analyses. I'll come to it later. So what was nice was that we was just doing it for on the side, this work, and we had no clue, I mean, how useful this work is. This was all a side project. All the students who were working on this project were doing it on site. They had their main projects. So when we published this, I mean, this is not that important. We got some amount of media attention, online, television, newspapers. But what was good about this attention is that, see, I'm not a chemist or a phytochemist or whatever. So, it's important to talk to the stakeholders for improvement of a resource so that the resource could be useful for the, different people who may potentially utilize it for their purposes. So after the media attention, what happened is that I could talk to various people, or they came forward and they told me where they would like improvements, what they like, what they would like improvements in. But we were essentially a small team. So based on the information or the feedback that I got from various people at that moment, and our group decided to essentially embark on a much more serious effort to both improve and expand the resource. And this was a very big effort, and this was led by my former PhD student Vivek Anand, who is now a postdoc with Nathan Price at Buck Institute for Research in Aging. And he essentially led this effort for about 4 years to make Impact a much more better database as it stands today. So I'll quickly summarize what we achieved over the next 4 years. First thing I would like to mention is that we realized that the high quality information happens to be in books, and those books were not digitized. It's just not scanning, but you have to synthesize the information from a paragraph and map the names to structure and so on. So what we did is that we actually digitized more than 100 books. Okay. And to give you an idea of the number of pages in these books, which are listed in the supplementary material of the paper, it is 83,000 pages. Okay. So we digitized the records from these books. Okay. The other thing we worked on, which required a lot of manual effort, is that, of course, as you know, if you look at the phytochemical content of a plant, it varies from different parts, from the leaf to the root, it varies. Okay. And it's important to document the known information at the level of plant part. So we had to revisit every data point which had been collected in Impact to map it to the plant part to which the phytochemical content had been associated. Essentially, we had to, for example, go back to all the 7,000 papers from where we had got the data in first version and get the plant part map. So we have standardized the information to 27 plant parts in our database. So what we further did is, of course, given the increase in the number of books, actually it was only 10 or 15 books in the first version, and now we have 143 books. So in the second version, so that led to a huge increase in the number of plants we covered. So we have finally information for 4,000 plants, so which was more than a twofold increase in the number of plants we have. And in terms of number of number of phytochemicals also, it's about a twofold increase. And in terms of number of associations, it is fivefold. So the number of plant part phytochemical associations we have is close to 1,90,000. So lastly, very important is that uh, you know today the all the scientific data which is of high quality uh, is ideally adhering to these two principles. These are known as FAIR and trust. FAIR stands for findable, accessible, interoperable, and reuse, and trust stands for transparency, responsibility, user focus, sustainability, and technology. So we worked tirelessly to ensure that Impact to adhere to both fair and trust principles. So this is among the very few data sets actually in India amongst the biological databases which adhere to these two principles. So let me just quickly walk you through the enhancement and expansion that we did. In terms of medicinal plants information, so of course we expanded the plants. So we have currently a database of roughly 7,000 non-redundant plants which are used in different systems of traditional medicine or even local health traditions. But the known phytochemical content, which is listed across these books or research articles that we cover, is only for 4,010, which is captured. In addition to the list of plants, there is a huge amount of other information that we have. For example, in which system of traditional medicine they may be used, what is their conservation status at the moment. We have also linked out to a digital herbarium in our database. So, I mean, the plant annotation is pretty detailed. But, as I said, our focus is always on the phytochemicals, that is the natural product. So we also worked hard to improve the annotation around the phytochemicals. So first is that, as I mentioned, we have all the association now of the phytochemical to the plant at the level of plant part. Okay, that was the major thing we did. We also worked hard in creating a non-redundant in silico phytochemical library where stereochemistry is properly resolved. Okay, so essentially this is a data set which is, I would say, stereo aware in that sense. So we have 1797967 phytochemicals in impact. You can ask me, like, in the first version we had 30,000 common names, of which only 15,000 could be mapped to a structure. Currently, we may have about roughly 80,000 to 90,000 common names, of which only 30 or 40,000 could be mapped. So there are about 40,000 names which we have picked up, which I call as strings in computer science, and we have not been able to associate to a structure. In Impact 2, we also did the following, that if the phytochemical was listed in that authoritative source and had a structure drawn, but let's say that chemical structure was not listed in PubChem or CAS or any databases, we did the hard work of actually drawing the structure by ourselves and putting that into Impact, okay, in a digital fashion. So all that effort was done. So it's not that these 40,000 names have a structure in the book and we have not captured it, but actually they've just named the compound, and it's not clear actually by doing basic chemistry what the structure is. So we don't put it out there. Lastly, there are two other enhancements that we have done. One, we have taken some high-end chem informatics approach to compute the molecular scaffolds of all these compounds at three different levels. I won't go into the details of that. And we have sort of predicted potentially the biosynthetic pathways of these phytochemicals. Essentially, I came from a metabolism perspective. And what has been bothering me, and I will end the talk, towards the end of the talk, I'd like to talk about it, that I mean, it's out in the open how many of these phytochemicals that are listed in impact we could find the part to some precursor in primary metabolism. Okay, that's a pretty much an open challenge. I believe we will be only able to do maybe for 10% of them at best with a lot of hard work. So if you are sort of wondering what are the books that we have digitized so far, so again, I want to mention that we have only limited ourselves to English language books. Okay, so there's a lot of vernacular books which are out there. That's beyond the scope because we are a very small team. But essentially, we have covered most of the authoritative books. So the Wealth of India, it was published in 1960s. When this was published, actually, it was covered in Nature as an important resource. This is a very important resource, Compendium of Indian Medicinal Plants, 6 volumes by CDRI. All the data has been captured. The books by Asima Chatterjee, um, the ICMR volumes, the pharmacopoeias. Okay, so the comprehensive list of books that we have digitized is in the supplementary material. So all these books are already covered in the Impact version 2. In terms of the therapeutic use, again, we went back to a expanded list of books which were authoritative, as I showed them, and we captured the therapeutic uses of these plants. The only major uh, enhancement that we did is that we have also the ICD-11 codes of these, therapeutic uses of these plants in Impact 2 as well. And also, as I mentioned, we have now the information of the therapeutic use also at the level of the parts. Okay, so how much time in would I have? Okay, so good. So essentially, Impact is accessible through this stable link. Also, it's mirrored at impact.com and impact.in. It's a very user-friendly database where you can browse the information by plant, therapeutic use, or phytochemical. You can also do various searches, and this advanced search link can be used to do some very rudimentary computations as well. So, I should mention that I have long-standing interest in secondary metabolism because I was working in fungi before I came to India. So we have also created a specific resource for secondary metabolites in fungi as well. And how we are exploiting them, I will tell you on another day. Okay. So, so since we have this large library of phytochemicals, there are certain amount of computations that we have done and we have put it out there for you to use. So one of the basic questions is that how can we divide into the different chemical classes, the phytochemicals that are compiled in, you know, impact. So it was very difficult to do it manually. So we use a tool called classifier, and the tool is used because this is the same tool which is used by drug banks to classify the all the FDA approved drugs. So if you look at it, the major classes are lipid and lipid-like molecules, phenylpropanoids, polyketides, and so on. Okay, in impact. We also use another tool called NP classifier. This is sort of a tool. It is sort of in an experimental stage, I would say, which tries to predict potential biosynthetic pathways which could be producing the uh, phytochemical of interest. Okay, but by no means it gives you the pathway of production. And finally, we also have this kind of a score which is given by this tool called NP likeness, which tries to see how natural product like the phytochemical is it. So, yeah, all this information is captured in impact. So this is an interesting analysis that we did in both version 1 and version 2. I'll talk to you briefly about this, and this leaves you with an important insight from the perspective of drug discovery. So this is a paper by Stuart Schreiber's group, who used to be the head of the broad drug discovery program. And in a couple of PNAS papers, what he did was very amazing. I like to summarize this in one slide. So essentially, they had three collections of compounds. One, which is a list of commercial compounds, roughly 6,000 compounds. Another list of about roughly 6,000 compounds, which is known as diversity-oriented synthesis compounds. This chemists use in drug discovery pipeline. And they had a set of natural products, roughly 2,500. So this 2,500 molecules has only a overlap of about 200 molecules with impact. Okay. So it's not that it doesn't have any overlap with impact, but about 200 molecules are common. And they characterize these sets of, molecules or compounds in terms of two quantities. And these quantities are very simple. One, which is known as stereochemical complexity. It is nothing but they count the number of stereocentric carbons in your molecule, okay, divided by the number of carbon atoms. Okay, so it's a number between 0 and 1. And second is number of carbons which are sp3 hybridized. Both quantities they defined in a way that it is doesn't depend on the size of the molecule. So for any molecule, these two numbers, stereochemical complexity and shape complexity, it's a number between 0 and 1. Okay, and what they did is that they basically plotted the distribution of these two quantities for these three sets: commercial compounds, diversity oriented synthesis compounds, and natural products. Okay, so this was the distributions they had. So if you try to do the mean and median of these distributions, you can convince yourself, and it is very clear, is that in terms of these quantities, the natural products have much higher values compared to commercial compounds, which are typically synthetic, and diversity oriented synthesis compound. So they didn't stop here, but what they did was very amazing in this paper. So they took a random assay of 100 proteins and they did the actual experiment of checking each of these molecules how many proteins they could bind to. And they come to a conclusion that typically natural products bind to much fewer molecules than commercial compounds and diversity oriented synthesis compounds. So I'm trying to summarize a large body of work in one slide. So essentially, they were finally able to correlate this specificity of binding to the values of stereochemical and shape complexity. Okay. So of course, we can't do the experiment. We are a purely theoretical place, but we decided to see if we take the complete set of phytochemicals in impact database, how is the distribution of these two quantities comparable to the sets they analyzed? So I basically turn their data into a box plot, the histograms. So if you see the commercial compounds, diversity oriented synthesis compounds, and then natural product set, how different they are in terms of the stereochemical and shape complexity. And if you analyze the 9,596 molecules in impact 1 and 17,967 molecules in impact 2, the distribution of these two quantities is very similar to the natural product set. However, the overlap between these two is only about 200 molecules. So, with this observation, of course, we can't do the experiment. One could potentially extrapolate and speculate that, like the natural product set analyzed by Stuart Schreiber, uh, it is possible that the phytochemicals that we have in our library are also going to be likely to be very specific binders of their targets. So, if you are planning to have a drug designed over a phytochemical, they will have be very specific to their targets and will have very few off targets. Okay, that could be advantage of going for natural product based drug discovery versus a synthetic molecule. And that was basically the message actually Stuart Schreiber left with this paper. And actually, in a subsequent paper, he analyzed 6 other physicochemical properties distribution for these 3 sets that he analyzed. We have also done the similar analysis for all the 6 plus 2 quantities. And what we show is that it doesn't matter what is the molecule, but as long as you have natural products, they have very similar distributions in these physicochemical properties. And we have also analyzed for our secondary metabolites in fungi, other databases. So this property seems to be universal for natural products. So I will skip this because this is very, very technical. So I just want to mention that if you want to do any of these computations, so we also have an open uh, GitHub repository of my lab, okay, where the codes for these computations are made public. So they use the open source tool RDKit. So if you have your own compounds, if you have the 2D structure in some SDF file, you can play around with them using our tools as well. So all the codes have been made public also on my GitHub page. So one of the important things we also did in the version 2 of the database is that we wanted to also be able to give an idea to the user on the different types of scaffolds out there. So without going to the detail, so we actually took the best state of the art tool in identifying scaffolds, and the scaffolds could actually be identified at 3 different resolutions, one at the graph level, graph node level, and graph node bond level. This is the highest resolution, doesn't matter. And so we computed for each of our 17,967 molecules, the scaffold, and the scaffold information at these 3 levels have been made public in the database. And what was more important was to see how diverse is our set. That is, you could have 1700 molecules, but they may have the same scaffold only. So what is the diversity of the scaffold in your chemical database? And we compared, for example, IMPACT2 with many other natural product and the complete PubChem database itself. And what we find in one word is that IMPACT has actually very high scaffold diversity compared to also other in silico and other databases that we have. So all this is actually published, so you're welcome to go and read it. And these are some of the frequent scaffolds that you see across phytochemicals in IMPACT. Of course, I omit the benzene ring, which is the most predominant and so on. So all this information is out there. So for example, in IMPACT, you can go and can actually search or select phytochemicals by a particular scaffold. Okay, so finally, we tried to also gauge through very preliminary computational analysis what could be the potential drug-like space of the phytochemical space that we have compiled. So, of course, I'm not an expert there. So, when you're not an expert, you can take the approach of wisdom of crowds. So, my student was an expert in cheminformatics. So, he actually used 6 different cheminformatics measures. I mean, these are based on different physicochemical properties, uh, based on which you try to predict which chemical could be potentially drug-like. So I think this analysis should be taken with a pinch of salt because many of the FDA-approved drugs fail some of these drug-likeness measures as well. But to cut the story short, of the 17967 molecules that we have, there are 1335 molecules which pass all the 6 drug-like filters that are used in the initial phase of the computational pipeline for drug discovery. And of these 1335 molecules, 11 are already FDA approved drugs. Of course, some of are sourced from natural products. And however, if you look at 90% of these drug-like phytochemicals, okay, this 1335 set, if you compare it with the small molecules approved by FDA to date, in terms of the chemical similarity and scaffolds, there is hardly an overlap. So in a sense, there could be new scaffolds and very different chemical structures which are out there to be tapped for drug discovery in our medicinal plant space, if I say so. Finally, of course, the reviewers were very interested in this. I mean, again, you should take this analysis with a pinch of salt because they wanted us to compare our data set with the phytochemical content of Chinese medicinal plants. Here, I should mention that neither impact is exhaustive nor any of the public databases on traditional Chinese medicine are exhaustive. But we had to do some analysis, so we took the one of the largest databases for traditional Chinese medicine or Chinese herbs, which is TCMH, because they give the mapping to the PubChem IDs of their phytochemicals. And we took the phytochemical set there, we compared it with impact. Roughly, there is about 20% overlap in phytochemicals at the drug likeness level or at the total phytochemical level. And there are scaffolds, of course, in impact which are not there in the sort of phytochemicals which are listed for traditional Chinese medicine. Okay, so to summarize, uh, as impact stands, this is where we are. So we have the plant along with its part and some information on the phytochemical content. We have the plant and the part with some information on the therapeutic effect. Currently in Impact 2, we don't have any information on formulations. Okay. But maybe in a year or so, we will have some high quality formulation data also available there, especially from an Ayurvedic perspective. And the question is, can we somehow link the cocktail of phytochemicals which are there in the formulations to the target proteins and explain perhaps the therapeutic effect? And so essentially, our long-term goal is to provide a state-of-the-art platform, which will be based on computational biology to sort of aid academics and industry if possible, working in traditional Indian knowledge and natural products-based drug discovery and wellness research. I should mention that, of course, Impact is heavily used for virtual screening. Okay. But the challenge is not to do just virtual screening, but actually take some of these leads and proceed to experiments. And there is hardly any serious effort towards that. I encourage colleagues who could do that to take it in that direction. So of course we also have screened against SARS-CoV-2, other viral infections as well. I mean, so that's clearly a utility of Impact. But I think it would be very important to actually take it to the experimental level. So that's in that direction. Very few studies are out there. So I think that is where I think that one should go towards. Okay, so of course, Impact is the largest resource on phytochemicals of Indian medicinal plants in India. But what is the state of the art on natural product resources for across the world? Okay. So here I would like to point out to this resource called Coconut. It stands for the collection of open natural products. This is not a manually curated database, but this is actually computationally created. What they have done is they have aggregated all the natural product databases, including Impact actually. So this version 2.0 just got published in NAR, Nucleic Acid Research. So you should be aware of this database. So they may be having about 500,000 natural product molecules out there. Okay, it's open source data. So all of you should become aware of that. And they also list all the resources on this database. So just type coconut and you will get it. Okay. So, and they list all the resources which they have compiled into a single resource. And of course, this is a fair and trust compliant database. Okay. Again, if they are unable to if they find a discrepancy with a particular molecule in a database, they don't put it in. Okay. They harmonize it properly. So essentially, because Impact was available in a fair and trustworthy manner, so they could also compile it into large. And this is not specific to plants; it is microbes. Yeah, so it's a huge resource. There is information also they have gathered from various sources which plants have been linked to some of these natural products. So the version 2 just got released. Lastly, a challenge, and this is where I think we want to go. And this is where omics would come uh, in actually. So, so Maya was presenting genome scale metabolic modeling of ginseng, right? But he particularly focused on a particular secondary metabolic pathway from primary metabolism. So if you look at it, there are 4,010 plants, medicinal plants, which are listed in impact. I would bet that not more than 20 of them have been sequenced to date. Okay. So when I was, coming to give this talk today, uh, so I also tried to think how many plant metabolic models have been built. So yesterday night, I was going through the plant psych, which is part of meta psych. Okay. So, so I tried to look for the plant with one of the largest phytochemical content known, at least listed in India, which is Catharanthus roseus. Actually, if you look at the metapsych database, uh, it's a it's still in process. Some group in US is still trying to actually annotate the metabolic pathways of Catharanthus roseus. Okay, that itself is in infancy. If you, I think Smita would present in her talk about her group's work on building the metabolic model of a medicinal plant. Okay, but then in that work also they use the genome of a related plant because they didn't have the genome of the plant they were building. And also the secondary metabolic pathway they built is particular to a particular phytochemical, not all secondary metabolic pathways. So essentially, you have two high quality or three at best plants with metabolic model. You may ask why. One of the simple reasons is that I may say on camera is that most computational biologists don't want to maybe venture into the plant space. So you have clearly Arabidopsis, rice, maybe maize, and they will also, I mean, there is also a challenge because you have to build the metabolic model by the tissue or the plant part as well. Okay. So that's clearly in infancy. And that true for primary metabolism of plants. Okay. Now we have, forget coconut. Coconut has, I mean, it's not specific to plants. We have about 18,000 phytochemicals, right? Now the question is how many of the 18,000 phytochemicals, which are secondary metabolites clearly, are covered in plant site? I don't know the answer, but I'll take a guess and say it is not more than 2,000. Maybe more like 1,000. So the question is that moving forward, can multi-omics sequencing uh, sort of mature to a stage that we could link these 18,000 phytochemicals to through a path, through a plausible chemical reaction pathway with known genes in some plant across the 4,000 plants that I've listed from the primary metabolite to each of the 18,000 phytochemicals. If we could achieve that, that would be a great, uh, scientific discovery, if I say so, because that could then be used for engineering microbes for potentially producing high value phytochemicals or so on. So I think we are far from there. We are doing very few pathways, and whatever is available is not typically available in these kinds of databases. So they have to be first compiled, and maybe we need also new computational tools to predict the pathways. So we may actually initially predict a pathway of set of reactions which may be linking from the primary metabolite to the secondary metabolite without knowing the genes or the enzymes. Then we have to use multi-omics to actually, predict the complete set of genes which may be doing it. And subsequently, you may then think of maybe engineering them in a microbe or so on. So I think we are far from here, and hardly anybody has attempted this in Indian medicinal plants. There is some attempts in TCM. There is hardly anybody I am aware of who has done that in India. In particular, if you're interested in India, I would like to mention that the only person who has made serious effort other than Smita actually in creating genome scale metabolic models is that by Sudeep Kundu in University of Calcutta. He was the one who actually with David Fell's group built the rice metabolic model for the first time. And he has also been pioneering on uh, incorporating omics for creating metabolic models for leaf, how light would influence the metabolic content in rice and so on. And very little has been done beyond that. For example, a question which emerges, which I was very keenly interested today, is that there is a lot of interest in, for example, in medicinal properties of food. Like medicinal rices are being seen from a new nutritional perspective. Can we actually use some of our genomic resources, because there is actually data for metabolomics available now for at least 30 or 40 varieties of medicinal rice in the country. Can we actually modify our metabolic models to capture those information at least? So we are far from there. And I think these kinds of courses would be very useful. I hope I could have attended and learned a lot. And so that would be the challenge to put forward. I don't know, we can't achieve it in Impact 3, but that's something we would like to do. That can we actually link each of our 18,000 phytochemicals what we have today to the primary metabolites through a pathway, which can happen in some plant or other. And I don't know how to do that actually currently. So thank you with that. Thank you for the opportunity.